So Chris is a third-year PhD student coming from South Korea. He did his bachelor's degree at Seoul National University. He was working on some more of materials research. And here, I think it's his real exposure to bio research, and he's been able to integrate his multidisciplinary training into solving biological problems. And during COVID, he had opportunities to actually delve into computational projects deeper, and this is one of two of his main computational projects, and this is to develop a software package that can help metabolic engineers or anyone who's studying metabolism quantify metabolic fluxes and free energies. So Chris, the floor is yours.
Thank you so much for introducing me, Dr. Park. I'm so glad to be here to have presentation of my past research, and my name is Keunseok Park, and I'm going to present a G-Flux, which is called a metabolic flux and free analysis software for interpreting stable isotope tracing data. So metabolism itself supports cellular energy production, redox homeostasis, and also synthesis of biomass production. So for example, metabolism synthesizes protein, lipids, and nucleotides, which are regarded as very crucial cellular building blocks. And also it generates ATP, which is a universal energy currency molecule in bioorganism. Lastly, it can also support cellular redox homeostasis by generating antioxidants molecule, for example, NADH and ADPH, to fight against reactive oxygen species, which is harmful for our DNA. So quantitative understanding of metabolism itself relies on rigorous measurements of metabolic flux. So what is metabolic flux? Metabolic flux is a metabolic reaction rate inside the cell. And what the researchers have been trying to measure the value itself was utilizing the stable isotope tracers, like the example on the right side. So if the cell uptake the 1,2,13C glucose, and this isotopes will travel around inside the metabolic network of interest. And then once we extract the metabolites, we throw out LCMS, we can get the mass isotopomer distributions. And by interpreting this isotope tracing data, we will be able to quantitate the metabolic fluxes with appropriate confidence interval range. For the past few years, 13C has been extensively implemented for resolving metabolic flux values. Besides 13C, deuterium and 15N can also provide more information into a metabolic network. So for example, if you look here, 1 deuterated glucose and 3 deuterated glucose can travel inside, oxidative pentose phosphate pathway to generate NADPH. And by utilizing this, tracers, we can resolve the fractional contribution of NADPH production flux in this case from oxidative pentose phosphate pathway. For 15N2 glutamine tracer, we are thinking that this tracer will be able to resolve the transaminase reaction reversibility because of the fractionally labeled glutamate measurement and 15N labeling pattern of essential amino acids. Besides measuring metabolic fluxes in the cell, another part of crucial field of metabolism is thermodynamics. So thermodynamics is about measuring the Gibbs free energy of metabolic reactions, and itself provides more actionable information. So I prepared a reaction diagram of certain metabolic reactions here. So if the metabolic reaction is strongly forward driven, then we can be readily more alter the pathway flux. However, if the reaction is close to equilibrium, we cannot actually change the metabolic flux easily. So how can we quantitate the Gibbs free energy of metabolic reactions? So conventionally, there are two ways of calculating delta G of metabolic reactions. The first one will be utilizing the equilibrium constants plus constant and the absolute metabolite concentrations inside the cells, which is very cumbersome. However, if we utilize the reaction reversibility, which is defined by reverse reaction flux divided by forward reaction flux, we can easily quantitate delta G based on this ratio. So also in part of isotope tracing experiment, how can we tell the reaction's reversibility based on substrate labeling pattern? So I prepared a chemical reaction where the substrates are labeled as red circle here, and then it detaches through this metabolic reaction. And in this reverse reaction, it gets the unlabeled solvent molecule from B to A to generate unlabeled substrate. So based on the observation of the labeling fraction of substrate A, we can tell how the reaction is far or close to equilibrium. So if the substrate labeling is very unlabeled, then reverse flux is very comparable to the forward flux, which means it's really close to equilibrium. If it's the opposite, then it's very strongly forward driven. So based on the need that we need to measure flux and free energy inside the cell, we developed a G-Flux that resolves the flux and free energy throughout multiple isotope tracer experiments. So this panel is a steps for operating our software to get eventually free energy and fluxes. So for example, if you're using, in your experiment, if you're using 1,2,13Z glucose, you can choose your type of isotope tracers here. And then if you're looking into upper glycolysis and pentose phosphate pathway, you can generate the metabolic models inside my software to build a model. And third, you have to generate you have to provide experimental data, which is a mass isotopomer distributions of extracted metabolites. And fourth, you have to define a flux constraint, which is needed because the software cannot, simulate all infinite range of fluxes. Now it's ready for computing the metabolic flux. So what it does is to minimize the simulated metabolite labeling pattern, to minimize the difference between the simulated labeling pattern and measured labeling pattern. And it finally turns out the metabolic flux in the end. And lastly, it computes the confidence intervals to tell us about the reliability of the computed fluxes. So the graphical user interface for G-Flux is looking like this. So as we had gone through the steps I explained in the previous slide, I prepared a steps for using our software. So if you are, for example, you used E. coli experiments and then 13C and deuterium, you can check the checkboxes correspondingly. And if you want to generate a glycolysis metabolic model, you can click glycolysis pathway here and click FBA reaction correspondingly. Then you can see the added reactions based on your selection. And once you build your required metabolic model here, then you are now ready for inputting your experimental data, which is going to be your identity of the isotope tracer, plus your measured labeling pattern of metabolites. So for example, if you look into this FBA reaction here, you must input the DHAP labeling pattern sequentially and with the biological replicates here. In terms of defining flux constraints, I need to introduce a new concept of fluxes. The first one is net flux. So net flux is just the subtraction between forward flux and backward flux. And then the exchange flux is the minimum between the forward and backward flux, which is more convenient for defining flux constraints. So we define these net flux and exchange flux, and we constrain the numerical value by inequalities. So graphical user interface, after clicking the compute metabolic flux button, you will encounter a result panels, which also shows the experimental data, which shows the labeling pattern of extracted metabolites, and also this finally computed fraction data here correspondingly, so you can compare the tendency of this. And furthermore, the core part of the software is to get the flux and also free energy. So we have this numerical table graphical user interface, which shows each individual reactions fluxes, and also finally you can see the free energy calculated on the very right. So that was the demonstration of how to quantitate flux and free energy via a G-Flux. But furthermore, we wanted to answer scientific questions via throughout our software, which is G-Flux, because G-Flux has another capability for simulating labeling patterns of metabolites through giving certain types of fluxes. So I prepared a question whether 18-O-glucose tracer can reveal the Gibbs free energy of glycolysis. So presumably the best tracer we have thought of is 6-18-O-glucose tracer. So in this metabolic model, we can see that it decomposes into GAP and DHAP. But because of the reaction reversibility of TPI, the DHAP could be also labeled by GAP metabolite. Also, because of the reverse reaction between labeled GAP and labeled DHAP, we can also observe the N+4 FBP labeling here. So based on the observation of DHAP labeling pattern and FBP labeling pattern, we thought that we are going to able to tell TPI reversibility and FBA reversibility. To further delve into how we can observe the Gibbs free energy of whole glycolysis, we simulated the labeling pattern of glycolytic metabolites here. So we can see that the labeling pattern decreases gradually along the reaction direction. So if you look into the final step of glycolysis, second to the last step, which is enolase, it detaches the labeled 18O here to make PEP unlabeled. So we thought that 6-18-O-glucose will be ideal for quantitating Gibbs free energy of E. coli. Another thing that we also have to consider is that not only just simulating the labeling pattern in a basal E. coli condition, but we wanted to test whether the labeling pattern of reversible glycolysis and forward-driven glycolysis differs each other a lot. So we can see that in the reversible glycolysis, the labeling pattern decreases in this behavior. But forward-driven case, the labeling pattern changes different from the reversible case. So we thought that within this range, the labeling pattern disparity will tell us about how glycolysis is close to equilibrium or like forward-driven. So we hypothesized that 6-18 would be ideal for quantitating the Gibbs free energy of glycolysis. So rather than relying on the simulated labeling pattern in the subsequent slide, we actually wanted to confirm that our software is able to actually calculate the same Gibbs free energy when we input the Gibbs free energy in our software. So I have prepared 4 different cases of glycolysis. The most left-hand side is extremely reversible, and the most right is extremely forward-driven. So based on this input Gibbs free energy, we generated a labeling pattern of metabolites correspondingly, and then we input that metabolite labeling pattern into the G-Flux to recompute this Gibbs free energy back. So what we have observed is that I prepared a XY scatter plot where the x-axis is the input which is the input free energy that we want to type into the software. And the y-axis is the regenerated, which is a computational value that G-Flux turned out based on the labeling pattern we give. So what I have concluded in this figure here is that we are able to get almost exactly the same Gibbs free energy until forward-driven case, which is whose Gibbs free energy is looking like this. However, if the Gibbs free energy of glycolysis is very strongly forward-driven, then there was some deviation between PGI and GAPD reaction. So we were able to say that in this tracer is able to answer the Gibbs free energy of glycolysis from reversible to forward-driven conditions. Another question that we also wanted to answer through the G-Flux is whether 13C and 59 isotope tracing is able to reveal transaminase and amino acid degradation reversibility. So this is the figure I also showed in the previous slides to once again explain there are a 15N2 glutamine to be substrate that gets into the cell. And then based on transaminase reaction, glutamate can be partially labeled by glutamine and also unlabeled nitrogen from amino acid, which is also uptaken by the cell. So based on observing the labeling pattern of amino acid in terms of nitrogen and glutamate, we thought that transaminase reversibility will be quantified. So that's the underlying principle that we thought of. And then for a glucose tracer, we also prepared glucose tracer because the degradation product of the amino acid is also the intermediate metabolites of TCA cycle. So for example, these 6 amino acids or more than this will degrade their carbons into feeding the TCA cycle by making acetyl-CoA, fumarate, and succinyl-CoA. But at the same time, glucose is supplying 13C into the TCA cycle. So based on the observation of acetyl-CoA, 13C labeling, fumarate, succinyl-CoA, 13C labeling, we hypothesized that we will be able to tell the amino acid carbon degradation flux with respect to glucose infiltration. So we had the simulation or the things or the thoughts based on our G-Flux. So we actually wanted to conduct a experiments to see whether 6-18 glucose is able to tell us about the reversibility of the glycolysis, and the tracing of this tracer will tell us about the amino acid degradation flux. So this 2 diagram is a schematic diagram of the experiments. For this case, this is E. coli, and this case is ACT116 cells, which is human colon cancer cells. So the reason for preparing these 2 different nitrogen replete, nitrogen limited case is that in 2019, Dr. Park realized that nitrogen source availability in E. coli rewired the glycolytic free energy. So we anticipate that nitrogen replete conditions glycolytic free energy. Glycolysis is basal or forward-driven. And if the nitrogen is limited, then glycolysis of E. coli will be close to equilibrium. For this mammalian cell experiment scheme, we prepared 2 different conditions where first one is basal reference, and the other is with the drugs. So what is the drug? The drug is inhibiting for amino acid uptake transporter. So we wanted to see whether our tracer is able to tell us about the amino acid degradation fluxes in 2 different scenarios. So that's the experimental scheme. So for the second experiment, we observed the labeling patterns of different amino acids and carbons also. But for this, I am showing the 59 labeling fraction under the drug BCH, which is amino acid uptake inhibitor. So based on this figure, So before going on to this figure, the x-axis is the fraction labeled of 15N in different metabolites under the amino acid inhibitor, uptake inhibitor, and the y-axis is the reference without any drugs. So we couldn't see like a substantial deviation between these two different conditions. So we thought that the drug concentration was not enough for inhibiting the amino acid uptake. So later on, we'll conduct another experiment for higher concentration of the drugs to see the difference of the labeling patterns. So besides this, labeling pattern, we actually wanted to see whether our software is able to tell us about the reaction transaminase reversibility. So we put that labeling pattern into our software, and then we quantitated the Gibbs free energy of amino acid transaminase. So actually, in this figure, you can see that serine, valine, methionine, and tyrosine reversibility was able to be quantified. So one takeaway from this figure is that valine and tyrosine transaminase is strongly forward driven, unlike the other two amino acids. So we were hypothetically able, we thought that like hypothetically to get all the other amino acid degradation reversibility, but somehow the software told us about only 4. So we are still working on like whether we are able to get the other transaminase reversibility based on like differing the model subsequently. So for the E. coli experiments, we assumed that we were able to get enough metabolic extractions, but in case of this experiment, we were not able to extract 2,3-DPG labeling, which is also substantial for calculating the Gibbs free energy of E. coli. So we will do the experiments again with E. coli to get the labeling pattern of anticipated glycolytic metabolites and calculate the Gibbs free energy further. For the ACT116 dual tracing experiments, we will do metabolic flux and free energy analysis with simple model. For example, for containing only transaminase reaction to find out our quantitation is doable. And if we confirm that our software is like simultaneously able to calculate the free energy of transaminase, then we will further delve into differing the drug concentrations to be higher to see the difference of the amino acid degradation fluxes. Yeah, so that's the future direction. I really want to thank Dr. Park for leading my research, and thank you for having me present in QCBio Research Institute. Thanks.